Dr. Joel Peterson is coming to us from the University of Wisconsin. That's the other UW, but it's okay, we'll let that slide. And he's the Rothamel Boscom Professor of Soil Science. He has appointments in the departments of Soil Science, Chemistry, and Civil Environmental Engineering. He's a faculty member in the Environmental Chemistry and Technology program and the Molecular and Environmental Toxicology Center. Professor Peterson's research group studies a variety of things, the physiochemistry and biophysical processes in terrestrial and aquatic environments. He has pioneered work in understanding the environmental transmission of prion diseases. The group's research emphasizes the mechanistic studies of the interaction of organic contaminants, micromolecules with environmental interfaces. Dr. Peterson has received some important awards. He's received the Career Award from the National Science Foundation and the Early Career Award from the Agronomy Society of America, the Crop Society of America, and the Soil Science Society of America. So coming to us from most of his education through California is now in Wisconsin, and we're hoping that he can take us the next step here in understanding the whole environment about what happens to this prion when it's out in the environment. Professor Peterson, can you come talk to us now?
Thank you for the introduction and for the opportunity to talk with you this morning. As was stated, I'm going to talk about soil and the environmental transmission of prion disease. Where is the laser pointer? Thank you. There's an on-off switch on the side. There's an on-off switch. Yeah, good. Thank you. Okay. But before I start talking about chronic wasting disease, I want to talk about a different disease for a few minutes, and that's sheep scrapie. Sheep scrapie has been around at least since the 1700s, and humans have coexisted with sheep that are infected with scrapie, which is another prion disease. And there's been no known transmission to humans. However, if you look back in the old agronomy literature, you see reports of shepherds commenting on fields being cursed. Whenever they had placed their animals out on those fields, they'd come down with this disease, scrapie. So the idea of environmental transmission has been around for a long time, more than 300 years. So there's good evidence for the environmental transmission of scrapie. One microcosm, if you will, is Iceland. Scrapie was introduced to Iceland in a shipment of sheep from Norway sometime in the 1800s, and then spread to a large portion of the island. And in many places, the farmers would try to eradicate their herds, repopulate them with sheep from other areas, and the disease would recur because of the existence of scrapie prions in the environment. A very interesting study was published in 2006, also in Iceland, where in one of these depopulated farms, sheep were reintroduced, but there was one portion of the farm that was where sheep were not allowed until 16 years after eradication. That's this area here around this house. And after 16 years, sheep that were housed in this area came down with scrapie. So at least in Icelandic conditions, scrapie prions can remain viable in the environment for at least 16 years. Another study out of the UK by Madison and co-workers examined the presence of scrapie infectivity on different structures within sheep environments, including water troughs, gates, penning, and feed troughs, and found that some of these could, from scrapie farms with scrapie, could transmit disease to other sheep. So not only may prions be present in soil, which is the bulk of what I'm going to talk about, but they can be present in other surfaces that are in the environment of sheep and deer. So one last look at one last bit of data from Scrapie. This is a study conducted in Germany by Björn Siedel, where he took Scrapie-infected brain material from hamsters, in this case, and put them, buried it in soil, and then periodically dug some up, and then measured the protein by Western blotting, which is a less sensitive technique than those that Davin was talking about. And even using these less sensitive techniques, after 29 months of interment in soil, infectivity still remained. Okay, if one would go back and repeat the study using some of the sensitive techniques like RT-QuIC and PMCA, I would expect that we would see prions surviving for much longer periods of time. A really seminal piece of work was done by Mike Miller in Colorado, where he demonstrated the environmental transmission of chronic wasting disease in mule deer. And in this case, he had 3 different experimental systems. In one set of pens, I believe it was 3 pens, he housed infected deers. In one set of 3 pens, he allowed infected carcasses to decompose. And then in one set of pens, he allowed infected deer to live, and then he removed them so the pens contained residual excreta. And then he placed healthy, or rather naive mule deer, and mule deer had never seen chronic wasting disease before. They were purchased elsewhere in the country. He introduced them into these pens and then waited to see if the disease would manifest, and indeed they did. In all 3 cases, the disease showed up in some of the animals. The prevalence is low, but the experiment was terminated when he began seeing chronic wasting disease in these animals, because these studies are quite expensive to conduct. So Davin introduced the idea of shedding of prions into the environment. Shedding can occur through feces, urine, and saliva. These numbers of infectious units means that one infectious unit is enough to kill half an animal population that's exposed. And these numbers are based on data in the literature and could probably be updated now that Davin has some new data. So once the prions are shed into the environment, they're subjected to a variety of processes that affect the distribution of the agent in the environment, its bioavailability, and its infectivity. And some of these processes include attachment to the surfaces of particles, soil particles, that can result in limiting their movement down through a soil column. It can also mean that prions associated with some soil particles may be moved by flowing water when we have severe runoff-producing rainstorms or with snowmelt. Some kinds of soil particles can actually inactivate prions. We've showed that, but I won't talk about that too much today. And then possibly also plant uptake. So once these prions are in the environment, they can be taken up by deer and elk via ingestion or through inhalation. And then these newly infected deer can begin shedding into the environment. Once the deer reach a terminal state, either of chronic wasting disease or they die for some other reason, all of the prions present in their body are introduced into the environment, and carcass decomposition is another route of entry into soil environments. A couple of other routes could be carcass disposal and release into wastewater treatment plants. So the bulk of what I'm going to talk about is interaction of prions with soil. So I think I need to give you a little bit of a background in soil. Soil is very heterogeneous at all length scales. So if you pick up a handful of soil, there are many different kinds of particles that are different shapes, sizes, chemistries. If you look across the landscape, you have different kinds of soil that differ in their relative abundance of different components. If you dig down in a soil, you'll see that there's different layers, and those also differ in composition. And because of all those differences in surface area, chemistry, et cetera, we can expect different degrees of interaction with prion protein. So what I'm showing you here are 4 kinds of components of soils that we have investigated. One is quartz sand. You see the scale bar here is 100 microns, so that's 1/10 of a millimeter. Kaolinite clay. This is a clay that's used for making porcelain, but it also occurs widely in the environment. These form these stack-like structures. Scale bar here is 10 micrometers, so 10-fold smaller than this. And then one that we've done a lot of work on, montmorillonite clay. This forms these flaky structures. The scale bar here is 1 micron, so 100 times smaller than these quartz sand particles. You may have heard montmorillonite occurs in deposits that are sometimes referred to as bentonite, which you may have heard of. There's large bentonite deposits in Wyoming. It's one of the major sources of montmorillonite clay in the U.S. And then humic substances are a component of the organic matter that's in soil. And organic matter, too, can have an effect on how prions interact with particles and how they and it may affect their infectivity. So just real quickly, have different components of soil, their chemical composition, specific surface area. So you see they vary quite a lot. Quartz, 1/10 of a square meter per gram. Montmorillonite, up to 800 square meters per gram of soil. So this is huge surface areas available for binding prions in the case of montmorillonite. And then these other properties are the cation exchange capacity, so the number of moles of cations that can be absorbed per square meter, moles of anions that can be absorbed, and then the pH at which the mineral has no charge. So at higher pHs, it's negatively charged. At lower pHs, it's positively charged. And so this varies. So mineral like quartz is going to be negatively charged. pHs in soil are typically 5 to 8 or so. So quartz is going to be always negatively charged. Some of these iron oxides may be positively charged over portions of the environmentally relevant pH range. David mentioned using iron oxides to clean up his samples. Iron oxides occur in the environment, and they can also bind proteins there, prions there. So a lot of what I'm going to talk about focuses on montmorillonite clay, so that abbreviation MTE you'll see a lot. Before I talk about soil, let's talk quickly about the charge properties of the protein, because prion protein is also charged. So this plot shows a measure of charge going from 0 into negative charges as a function of pH, and the prion protein is negatively charged over the pH range that we're interested in, and it becomes more negatively charged as the pH increases, as the soil becomes more alkaline, less acidic. Okay, and the charge depends on the form of the protein. There are different forms that are released into the environment. There's full-length prion protein and then a prion protein that's truncated at one end. And that truncation results in a more negative charge, that the part that's cut off is positively charged. Okay, so some of our early work focused on simply the binding of prions to different types of soil particles. And the data I'm showing you here are for montmorillonite and fine quartz sand. In the case, in these experiments, we simply mix the protein with the particles and then separate them by centrifugation. We measure prions in the liquid that remains after centrifugation and then in the particles. And what we find for montmorillonite, all of the detectable protein is associated with the particles. In contrast with fine quartz sand, all of the detectable protein is present in the free state. It's not bound to the quartz sand in this experiment. This was interesting. It was also interesting that you see these bands don't line up. One would expect the bands to line up. The position of these bands indicates the size of the protein. So somehow the protein is smaller when it comes off of montmorillonite clay. And so we examined this using antibodies that probe different parts of the protein. And when we use an antibody that probes one of the ends of the protein, the end that has the positive charges, we find that in the absence of montmorillonite, we have a signal. In the presence of montmorillonite or extracted from montmorillonite, there is no signal. So this indicates that this positively charged part is lost when we extract the protein from montmorillonite. And so this indicates, this indicates that part of the protein is involved in the interaction. We then went on and tried to remove the protein from montmorillonite clay using a variety of methods that others have used to extract other proteins from the exact same clay, and found that none of them worked. Okay, so we adjusted pH, low and high pH. In each of these cases, we have a primary extraction, which is what I indicate at the top, and then a secondary extraction, which is a denaturing detergent at 100 degrees Celsius, and that's effective. So extremes in pH don't remove protein, these blank lanes. Increasing salt concentrations don't remove the protein, blank lanes. Even compounds like urea and guanidinium hydrochloride, which are compounds that break up the hydrogen bonding structure of water and are usually effective in extracting proteins, they don't work either. So what does this all mean? This all means that in the case of montmorillonite, the prion proteins are interacting very strongly with the particles. Okay, that's very hard to remove, more difficult to remove than other proteins are from these kinds of particles. Having learned that, we expected that would reduce the bioavailability of prions when they're bound to montmorillonite. And so we did what prion researchers typically do when they want to assess the infectivity of a sample. We took the sample and we injected it into the brains of laboratory animals, in this case hamsters. And our expectation was that we would see a reduction or maybe an elimination of disease transmission with prions bound to montmorillonite. But we were wrong. Okay? So the first bar shows animals that receive no prions, and they're okay. None of them came down with the disease. Interesting. Animals that received just montmorillonite, none came down with the disease. Animals that were exposed to prions, all of them came down to with the disease. Animals exposed to prions associated with montmorillonite particles, all of them came down with the disease. In animal studies, the incubation period is a measure of the amount of infectivity present. So these data suggest that there is no reduction in infectivity in the presence of montmorillonite. I need to say one other thing about length of incubation. So length of incubation time is a measure of how infective the sample is. Shorter incubation times means more infectivity. Okay, so there's no reduction in infectivity when montmorillonite is present, perhaps an increase. Okay, so this is interesting. So attachment to montmorillonite does not diminish transmission via intracerebral route, and does not diminish via intracerebral route. But obviously, this is not environmentally relevant. A deer or elk that gets montmorillonite-bound prions injected into its brain in nature is going to die from something else in all likelihood. So we did the next logical experiment, which was to orally expose the animals, again with the same expectation that bound to montmorillonite, the animals would come down with the disease at a lower rate. And we were, again, wrong. Okay, so what I'm showing you here is a survival curve, days after inoculation, and then animals remaining on the y-axis. The black line are for animals that received only prions, and the red dotted line is for animals that received the same amount of prions but associated with montmorillonite clay. And what we find is more animals come down with the disease when the prions are associated with montmorillonite clay. And not only that, they come down earlier than the animals that receive only prions. And if we lower the dose by a factor of 10, this is a dose so low that none of the animals show clinical symptoms of disease. Even at that low dose, when montmorillonite is present, we again have disease showing up in animals. So the penetrance occurs at doses that fail to initiate disease when montmorillonite is present. We worked with a biostatistician to model some of these data using what's known as a Cox proportional hazards model, and he came up with a factor by which montmorillonite enhances disease transmission, and that factor was 680. There's some bounds on how good this estimate is. It's a 95% confidence interval. Lower bound is 16. The upper bound is rather large. We weren't able to estimate it, so it's infinities. We went on to examine other particles for their ability to potentiate disease transmission. So here I'm showing you similar sort of data: montmorillonite clay, kaolinite clay, and quartz sand. In this case, all the experiments are all done at prion concentrations that don't produce disease in animals when there's no particle present. So for all three mineral particles, there's a decrease. There's an increase in penetrance. Disease is initiated in the presence of the particles, where it is not in the absence of particles. And there's some difference. The montmorillonite seems to be perhaps the most efficient, closely followed by kaolinite, and then quartz microparticles are a bit less effective at enhancing disease transmission. And this has to do with the chemistry of the particles. We then examined the effect of whole soils on disease transmission via the oral route. So here we have prions in black again, and then three soils, and we see that there's a good deal of variability in how they impact disease transmission. In the case of the Dodge soil, there is no real difference from the soil-free exposure, but these two other soils increase disease transmission. This table at the bottom gives some of the properties. So this is the fraction of organic carbon, highest for the Dodge soil, the sample that did not affect disease transmission. The two other soils have low organic carbon contents. Clay composition is lowest in the Dodge soil that did not enhance transmission, higher in the other two. So we've shown, I think convincingly, that mineral particles and whole soils can both enhance oral transmission. There's a nice study that was published just last month coming out of Colorado State, Glenn Telling and Mark Zabel. And in this case, they took soil from deer and elk pens that had housed diseased animals, and they allowed mice to live in tanks that had the soil as substrate for about a year. These are special mice. These mice are transgenic. Their gene for the prion protein has been taken out, and the gene for elk prion protein has been put in, so it makes them susceptible to chronic wasting disease. And so they allowed them to live, and then they examined these mice after a year for the presence of prion infectivity using a sensitive technique, protein misfolding cyclical amplification. So the white bars are just acute exposure to PMCA is comparable to those that have been fed prions directly. They went on to do a couple of interesting further studies. They fluorescently labeled prions and then associated them with fluorescently labeled protein, prion protein, and in some cases associated with montmorillonite, and then they fed it to mice and then examined where in the mouse this fluorescence occurred. And so after 1 hour, they're seeing fluorescence in the prion exposed and prion plus montmorillonite exposed mice, not in the sucrose exposed mouse, fortunately, and the signal increases over time up to 2 hours and then decreases after that. They went in and examined the cells within some of these organs to determine how many cells contained fluorescent prions, and they found that there was an increase in the number of prions when montmorillonite was present in Peyer's patches, in small intestine, in the cecum, and in the spleen. Peyer's patches are small lymphatic tissue in the intestine, whose purpose is to sample antigens in the intestinal lumen. And so these can be pulling prions associated with montmorillonite into the lymphatic system. Okay, so there's good evidence that association with particles, particularly montmorillonite clay particles, impacts disease transmission, promotes disease transmission. A study several years ago tried to correlate the incidence of chronic wasting disease in Colorado with the clay content of soil. And in this case, clay content simply means a size fraction of the soil where most of the clay minerals exist. And this map was showing percent clay in a red to gray scale. So red is higher clay content, gray is low clay content. The dots are prion-infected deer. The X's are deer that aren't infected by prions, apparently. And they found in regressing these data that for every 1% increase in the fraction of clay in the soil, there was a nearly 9% increase in the odds that an animal would be infected with chronic wasting disease. And they adjusted their analyses for the age of the animals, the sex, and other features of the habitat. So those are excluded in this analysis. Okay. Okay, I had several slides like this, but I decided to remove them, and I just want to talk about one to illustrate a point. So I've talked to you about the charge properties of the prion and how it changes. It depends on pH, and also that the charge properties of minerals or particles in the soil also depend on pH. I'm giving you an example here of aluminum oxide. It's not a very common mineral in soil, but it proves the point. So it has what is known as a pH of 0 point of charge, a pH at which it is neutrally charged. It bears no net charge at about 7.75. So below that, in the blue, the mineral is positively charged. Above that, the mineral is negatively charged. Okay? And what I'm showing you is referred to as attachment efficiency. Just think about this as how much the prion wants to associate with the surface. And what I'm showing is that at low pHs, there's a large amount of attachment. Okay, when the mineral surface is positively charged, as it becomes negatively charged, there's less attachment. Okay? And these experiments were done at 2 salt concentrations, indicated by I here. So at high salt concentrations, there's more association than at low salt concentrations. And 2 forms of the prion, one that's full length and one that has the positively charged end cut off, and that also impacts the degree of attachment. So the one that lacks the positively charged end binds to a lesser extent than the one that has a positively charged end. Okay, so the points I want to make here are, well, one, that it's complicated, right? So we can't just simply say prions bind to soil. It depends on what kind of particle is present in the soil. It depends on what form of the prion is there. It depends on the pH of the soil. And again, pH of soils range from 4 to 9. It depends on how saline the soil is. Okay, so it's hard to make blanket statements. And much of the research to date has focused on just a handful of soil components, particularly this montmorillonite clay, but there are other components that haven't been explored that could well be important in some environments. How am I doing on time? 6 minutes, okay. Okay, so now I'm going to shift gears and talk a little bit about inactivation of prions, because when we have prions in the environment, either the natural environment or when we have animals in captivity, there may be the interest to decontaminate facilities or soils. Looking at the literature, there's a variety of techniques that have been attempted to inactivate prions, and I'll walk you through this table just quickly. So electromagnetic radiation like ultraviolet light, microwave, ionizing radiation, which is X-rays, these are not effective against prions. They're effective against bacteria and viruses, but not against prions. Chemical disinfectants, alcohol, ammonia, benzene, ethylene oxide, formaldehyde, these are all not effective at eliminating infectivity. Some rather extreme chemical disinfectants are 1 molar sodium hydroxide, a strong base, acidic denaturing detergent, and high concentrations of a chaotropic agent. In terms of oxidation processes that would oxidize the protein, there's a variety that have proven to be less than sufficient in eliminating infectivity. High concentrations of sodium hypochlorite, also known as bleach, can work as long as there's not a strong demand for the chlorine in the environment. There's been some work on biological degradation using, so for most proteases, these are enzymes that break down proteins, for activated sludge, this is in wastewater treatment plants, and anaerobic sludge digestion also in wastewater treatment plants. These are not effective means. There are some reports of proteases in combination with other conditions that are effective, so including detergents or elevating temperature, say to over 100 degrees, can promote degradation. And then heat, even heat is not always, heat does not inactivate prions in the same way it inactivates other organisms. Boiling and standard autoclaving isn't effective. One has to autoclave it under special conditions. So we've been investigating ways to inactivate prions. We've done several different kinds of study. I just wanted to highlight one, and this is using an oxidant known as peroxymonosulfate. It's used for shock disinfection of swimming pools, among other things. And so what I'm showing here is on the left-hand plot, the concentration of this of peroxymonosulfate, and the y-axis is the band in a Western blot, the band intensity relative to control. And so as we increase the peroxymonosulfate concentration, the amount of protein that is detected decreases. This works for hamster prions as well as CWD prions, and it's also time dependent. Working at 2 different concentrations here, we have a large amount of inactivation for the higher concentration by about 15 minutes, for both within an hour. And this degradation can be enhanced by adding a transition metal such as cobalt, and it occurs in the presence of competing biomolecules. If we've examined the reduction in infectivity. Also, I'm showing you just one blot from a series from study using protein misfolding cyclical amplification. And the short story is that we find nearly 6 log reduction, a million-fold reduction in the PRP-C to PRP TSE converting activity, so the way that these proteins propagate themselves. So this, we think, holds promise as a way of inactivating prions. We have studies nearly ready for submission to journals examining the effect of peroxymonosulfate on prions bound to soil particles. To finish up, I just wanted to highlight work of another researcher, Claudio Soto, down in Texas, who's been examining, we have been examining interaction of prions with plants, and he has as well, and he's published some work recently that I thought you might find interesting. So here he took wheat plants, exposed their leaves and roots to CWD prions in urine or feces, and then washed them thoroughly, and then measured using PMCA the prions that remained. And he found that small amounts of prions from urine associate with plant leaves and roots, small amounts from feces associating with plant leaves. He also grew plants in this case, barley plants grown in soil amended with pretty high concentrations of infected brain homogenate, but he did find that some of this material showed up after 3 weeks of growth in the stems of the plants as well as in the leaves of the plants. So some evidence that plants can also take up prions, and this is an area that's not been explored very much, but this could be another route by which animals are infected. So to summarize, prion association with clay particles enhances oral disease transmission. Naturally contaminated soils can lead to disease transmission. Nice work from Colorado State. Electrostatic interactions, I didn't use that word, but charge-charge interactions seem to dominate attachment to mineral surfaces and organic matter, shown that proximonosulfate can inactivate prions, and this may hold promise as a surface decontamination agent, and plants can take up prions and translocate them to above-ground tissues. And to finish, I'd just like to thank all those that have contributed, people in my group, my collaborators Judd Aiken and Debbie McKenzie, who were at UW-Madison and have since moved to University of Alberta, and then a variety of others, including Glenn Telling, who provided us with transgenic mice. So with that, I'd be happy to take any questions.
You mentioned that the disease has been present from about 1700s. Is there an idea where it originally came from? What was the process? How did these prions develop? Was this some kind of a mutation is there a possible source, and do we know anything about this?
So the question was, how did prion diseases arise, given that we know, at least for Scrapie, it's been around for centuries, since the 1700s. The answer is no. There's not a mutation involved in most prion diseases. There are a few in humans where a mutation is involved, but in most cases, there's not a mutation involved. It's not clear why these diseases occur. It's been hypothesized that in the brain, in the nervous system of animals, there's a equilibrium between different folding states of the protein, and that in some cases, for reasons we don't understand, the equilibrium shifts to this misfolded form, and it gradually accumulates in the nervous system. We really don't know.
You really caught my attention when you said that, I believe you said that CWD can be found in wastewater treatment plants. And I'm just wondering if I did hear that correctly, could you just touch on that a little bit and what has been found and what does that mean in the bigger picture?
Okay, so the question was, can CWD agent be found in wastewater treatment plants? I included wastewater treatment plants on a slide to indicate that is a route of entry into the environment that people have expressed concern about. There is no data suggesting that CWD proteins are present in wastewater treatment plant, influent or effluent. It's certainly possible that through slaughterhouses with mad cow disease, for example, might emit prions into wastewater treatment plants, but we don't have any data on that. My group has done some work on studying the persistence or the survival of prions within wastewater treatment plants. They're not inactivated in conventional wastewater treatment processes. There was one report several years ago by Tracy Nichols in that what appeared to be a single detection of prions in water entering a water treatment plant not a wastewater treatment plant, a water treatment plant, drinking water, in Colorado. And this seems to have been linked to runoff from CWD, in the area where CWD is prevalent. It was not detected in the water exiting the plant. Yes? Or
I guess that might raise some concerns concerns here with, for example, the elk on the National Elk Refuge reside above the town of Jackson's water supply. Would you expect that infectious proteins would be bound to these soils, or is there any evidence of, kind of percolate through the soils to the water supply?
I haven't examined the soils there, so I don't know the composition. Our studies using a variety of soils and examining leaching through the soil column suggests that there's relatively limited vertical movement of prions within the soil. Binding to particles could be a route by which they're washed off land surfaces into surface waters, streams and rivers. Water treatment plants have processes to remove particles. When you turn on the tap, you don't have a bunch of sediment in your drinking water normally. So there are processes to remove those, and they should be effective against prions that are associated with particles, although that has not been studied.
Do we have information on the level of the dose, the quantity of the dose, how that impacts the risk of infection? I mean, you have a plant that's uptaking prions through the subsurface tissue versus a heavily applied application in a confined space. How much does the amount of dose impact infection rate?
The question is, how much does dose impact infection rate? With a specific concern about the uptake of prions into plants. So there is a very strong correlation with the amount of prions that an animal is exposed to and its likelihood of contracting disease, as well as how rapidly the disease, how soon disease symptoms are manifested. Those relationships are best understood in laboratory animals like mice and hamsters, less well understood in deer and elk. The levels of prions in the plants in the study I showed you, they were not able to quantify in that study because of some difficulties with their detection method, so I can't really say too much about that. They weren't able to say very much about that. It's something that my group is studying and something that we will have more information about soon. That's a very good question, and one we had thought about. I'm sorry. The question related to the peroxymonosulfate as a potential prion inactivation method for soils and what effect that chemical would have on the soil itself. And so it's a good question, something we've thought about. We've done experiments where we've examined the fungal and bacterial communities after exposure to this oxidant, as well as the ability of soil to support the growth of grass. And we find that there is some alteration in the microbiology of the soil, but it seems to recover, and the soil can support growth of grass. The chemical is very reactive, so it doesn't stick around in the environment very long.
Was UV light also included in terms of the microwave treatment?
Question was whether UV light was considered as a prion sterilization or disinfection measure. It's been examined in the past. It was one of the first things to be examined, and it's not effective.
How might prion association with soil particles affect its longevity, uterus clay and sand?
That's a good question, and one that's a little hard to get at directly experimentally. We think it will association with particles would enhance the longevity because it would reduce the access of proteases from microorganisms or chemical oxidants to the protein.
sure as you know, are often added to ruminant feeds to promote ruminal bypass of protein. Could that sort of process play any role in enhancing infectivity in mule deer milk? A protein bypass because of plant proteins are, and presumably prions would be bound to proteins, therefore they wouldn't undergo ruminal digestion, would pass into the...
I see. So did I understand you correctly that the question is whether association with proteins, prion protein with clay particles would prevent them from being degraded in the rumen?
I mean, I'm just thinking that adding clays to feeds for ruminants is commonly done
Yep.
as a technique to enhance protein digestion by getting proteins to bypass the rumen.
Mm-hmm.
And if any breakdown were going on prions in the rumen, then binding the clays would perhaps make them more effective. Have you thought about that?
We have, yes, we have thought about that. We've work done at University of Nebraska has shown that there's not too much degradation of prions when they go through the rumen, or at least simulated rumen fluids. But I would agree with you that association with particles should reduce or eliminate any degradation that might occur. So that may well be an important factor with deer and elk. The data I showed you on oral transmission from naturally contaminated soils was with mice, and their digestive system is quite a bit different than that of deer and elk.
I think we're out of time.